Hello everyone, we're going to talk about quality control of liquid dosage forms. Liquid dosage forms are potable pharmaceutical formulations which contain a mixture of active drug components and non-drug components like excipients dissolved or suspended in a suitable solvent or mixture of solvents. Liquid dosage forms are pharmaceutical preparation designed to provide maximum therapeutic response in a target population with difficulty swallowing the solid dosage forms and also to produce rapid therapeutic effects. Administrations and formulations. Liquid dosage forms can be supplied as ready-to-use liquids or powder for reconstitution. They are administered by oral, parenteral inhalation, ophthalmic, otic, nasal, and topical routes. Oral liquids are homogeneous liquid preparation that usually contain solution, emulsion, or suspension of one or more active ingredient in a suitable liquid base. Quality control is an essential operation of the pharmaceutical industry. It is the monitoring process which encompasses specifications, inspection, analysis, and recommendations. The drug and another pharmaceutical material utilized must be compatible with one another to produce drug products that is stable, effective, potent, easy to administer, and well tolerated. Advantages of liquid dosage forms. Oral liquid dosage forms are the most suitable dosage form for patients who have difficulty taking tablet or capsule, as in case with pediatric or geriatric patients. Drug with bitter and unpleasant taste can be given in sweetened, colored, and flavored vehicles. Drug with large doses can be easily administered as liquid dosage form. The faster absorption and distribution of the liquid dosage form than solid dosage form. Liquid dosage forms are more economical to produce than solid dosage forms. Disadvantages of liquid dosage forms. Liquid, especially aqueous preparation, are susceptible to microbial contamination. Masking the unpleasant taste of the drug in a solution is more difficult than with the drug in a solid dosage form. Administration of the correct dose is less precise since it depends on the ability of the patient to measure the correct dose using suitable measuring device such as spoon or a dropper. Liquid dosage forms are less stable compared to solid dosage forms like tablet or capsule. Pharmaceutical liquids are packed in a glass container, which are prone to loss by break of the container.
Type of the liquid dosage form. Liquid dosage form is divided into two types, monophasic and biphasic. And the monophasic is divided into two types, aqueous and non-aqueous. And the biphasic is divided into two types, suspension and emulsion. Type of liquid dosage form included in this presentation. 1. Solution. Oral solution are oral liquid preparation containing one or more active ingredients and excipients dissolved in a stable vehicle. Water is the most commonly used as a solvent. Also, organic solvents are used in the combination with the water or on their own. 2. Suspension. Suspension is a heterogeneous mixture containing solid particles that are significantly large for sedimentation. A suspension consists of a dispersion of relatively coarse particles, usually in aqueous vehicle. Suspension may be used for oral or topical administration. Oral suspension are useful in children and patients who cannot tolerate solid dosage form. 3. Emulsion. Emulsion is a mixture of two or more liquids that are normally immiscible. Emulsion are two-phase system, consist of liquid drug substance. They are classified as oil in water emulsion and water in oil emulsion. Emulsion can be administered orally or topically or intramuscular.
Tests for liquid dosage forms. There are general quality control tests that apply to all types of liquid dosage forms, such as visual inspection. For oral solutions, they should be clear and free from any precipitate. If discoloration or cloudiness is present, that may indicate chemical degradation or microbial contamination. And for oral suspensions, physical instability manifested by the appearance of flocculants or sediments that do not easily disperse with gentle shaking. Next, for emulsions. Phase separation that is not easily reversed on gentle shaking indicate physical instability. Next, we have microbiological tests, in which we assess microbial contamination through the viable aerobic count. The tests provide a quantitative enumeration of mesophilic bacteria and fungi that may grow under aerobic conditions. According to the United States Pharmacopoeia and National Formulary, the product is considered compliant with the test if there are no colonies present or if the results of the identification tests are negative. Another essential quality control measure for pharmaceutical liquid dosage forms is viscosity test, which refers to the resistance of liquid to flow, and this is a critical parameter in ensuring the proper administration, stability, and overall quality. Viscosity is measured by using a viscometer. There are different types of viscometers that are used based on the specific characteristic of liquid. pH test. The value of pH represents the acidity or alkalinity of an aqueous solution. The pH must be optimum as for oral liquid preparation as they are administered. It should be measured to ensure that it falls within the specified range, since it can affect the stability and solubility of the drug. To measure the pH value of a solution, this is done potentiometrically for more accuracy by means of glass electrode, a reference electrode, and a pH meter.
Specific test. We have specific test for suspension and emulsion. First test for suspension is sedimentation volume. Is the most important parameter in evaluation stability of suspension. Sedimentation volume is a ratio of ultimate volume sediment VU to the original volume of sediment VO before settling. The sedimentation volume gives only a qualitative account of fluctuation. Sedimentation volume F equal VU over VO. The F value is between the limit 0 to 1. The larger this ratio, the better is the retraceability as we see in this table. The second test for suspension is degree of fluctuation. It is the ratio of sedimentation volume of fluctuated suspension F to the sedimentation volume of defluctuated suspension F infinite. The minimum value of P is 1 when fluctuated suspension sedimentation volume is equal to the sedimentation volume of defluctuated suspension. The third test is redispersibility. Describe the ease of redispersion of formed sediment by moderate shaking to yield a homogeneous system. Ease of redispersion of the suspension can be determined either by simple agitation of suspension in container, the use of mechanical shaking device which stimulates a human arm motion during the shaking process and can give more reproducible results. Specific test for emulsion. The first test is phase separation. An approximate estimate of phase separation may be obtained visually. In general, creaming, fluctuation, and coalescence have occurred before phase separation is visible, this sometimes making quantitative evaluation more difficult. To measure phase separation, the rate and degree can be easily determined by keeping certain amount in graduated cylinder and measuring the volume of separated phase after definite time intervals. The second test for emulsion is droplet size. The growth in droplet size after preparation of emulsion is indicating of its physical instability. The droplet size is measured by microscopic method or by electronic device. The viscosity increases with decrease in droplet size because the smaller droplets tend to be more rigid. The third test for emulsion is thermal stress. It's usual to evaluate the stability of emulsion by subjecting it to high and low temperature in alternating cycle. The samples are first exposed to 60 degrees for a few hours and then to 40 degrees. Such exposure are repeated number of times and emulsion stability assessed after each cycle. Zeta potential is a specific test for both suspension and emulsion. It has practical application in the stability of systems containing dispersed particles, since this potential governs the degree of repulsion between the adjacent similarly charged dispersed particle. It can be determined by observing the movement of a droplet under the influence of an electric current.